Our next topic is going to be describing the four types of molecules that comprise the body. It's a little bit confusing because down the line we're going to be talking about the four tissues that comprise the body. This is a little bit different here. We're only talking about categories of molecules. Every single thing that your body is made of, is comprised of, the composition of your body, all of it fits into one of these four types of molecules. Before we begin that, though, it's important to discuss two types of reactions. And I'll be giving lots of examples of this. It kind of won't make tons of sense at first. But as we give more examples of this, hopefully it'll start to make sense. Okay, so first of all, we talk about a dehydration synthesis reaction. The dehydration means the removal of water, hydro meaning water. Synthesis means putting two things together. So in this picture, what we're showing here is two monomers, which are going to make a dimer. Mono meaning one, and dimer meaning two. So we have the two monomers are going to come together to be linked. Now, in order for that to happen, you have to have oftentimes some kind of an enzyme or something like that involved in the process. But for sure, what you're going to have is the removal of a molecule of water. And again, I'll show examples of this so that you'll kind of be able to follow along. But in a general sense, if you have two OH groups that are hanging off of here, and this is the joining part, what's going to happen is these two monomers are going to come together and that water is going to be removed. One hydrogen here, and one oxygen here. And so we're left with just the one O left over, and that ends up linking the two. So that is a dehydration synthesis, the removal of water in order to combine two things together. The opposite is true when you want to break two things apart. Oftentimes, again, this does require an enzyme to catalyze the reaction. Here, we start off with a dimer, two structures bonded together. There you see your O. And what you have to do in order to break these apart is you have to add water into the reaction. And then they're able to be broken apart. And here is right here is the O that was shown here. It is now attached to an H. And then here's the other OH here attached to monomer 1, if you will. The word hydrolysis, again, you see that hydro, which means water. The word lysis means breaking apart. Hydrolysis reaction is breaking two structures apart in the presence of water, using water to catalyze the reaction. That being said, we're going to start off describing our first type of molecule. We're going to be starting off with a carbohydrate. This is the full structure. As you can see, there's lots of carbons here. This would be considered a hexamer or a six-sided sugar. Carbohydrates would be our sugars. And here's a simplified structure. Oftentimes we'll denote it as simply a ring. If you see a six-sided ring or a five-sided ring, a pentamer, five-sided ring. Even if you don't see all the carbons in there, it's very likely that structure, at least in this class, it's very likely that structure is a sugar or a carbohydrate. All right, in your body, there's 5 different types of monosaccharides. Our carbohydrates are categorized by saccharides, monosaccharides, disaccharides, and polysaccharides. Mono meaning one, di meaning two, poly meaning many. So there's 3 types of monosaccharides we're able to consume: glucose, fructose, and galactose. Those are things we can actually absorb into our body, and they're easily able to give us an energy source. We also have 2 sugars over here, deoxyribose and ribose. These are important for the composition of nucleic acids, DNA and RNA. In fact, this is where the words DNA and RNA come from. Deoxyribose is a component of DNA. Ribose is a component of RNA. When we combine these things together, remember this would be called a dehydration synthesis, you would end up with combining them into different structures, ending with some sugars that you may have already heard of before. For example, lactose is the sugar commonly found in milk. This is a dimer that is created of 2 different monomer sugars or monosaccharides, galactose and glucose. There they are, galactose, glucose. Sucrose is made of a glucose molecule and a fructose molecule. And so those are the disaccharides that we commonly have as part of our foodstuffs and that we will break apart and probably consume and perhaps break apart for energy. At the bottom here we have polysaccharides. This is many sugars that are attached to each other. These have different roles in the body. You'll see them hanging, for example, off of a protein. This would be called a glycoprotein. You see the word glycogen there, which is oftentimes used for a complex sugar. If you have something like this hanging off of a protein is called a glycoprotein. If you have something like this hanging off of fat, it would be called a glycolipid. And so it has different roles like that. So in a general sense, carbohydrates would be a group of molecules including sugars and starches. They contain carbon, hydrogen, and oxygen. The major functions of carbohydrates in the body is to provide cellular fuel, especially if you're looking at these three guys right here, and you notice that they end with the OSE there, OSE meaning sugar. And then these guys as well. These provide sugar for us as well, but only when they're broken apart through a hydrolysis reaction. The monosaccharides are simple sugars that are single chain or single ring structures. Disaccharides are formed when two monosaccharides are joined by dehydration synthesis. And polysaccharides are long chains of monosaccharides linked together by dehydration synthesis.